All right, good morning, good evening, good afternoon, wherever you may be. Thank you very much for joining our webinar today. This is the 8th webinar in our webinar series, and we're very excited to have with us today Dr. Murat Akcakaya. Dr. Akcakaya is from the University of Pittsburgh and the Department of Electrical and Computer Engineering. He's going to be talking to us today about a recent publication that he and his team produced in Scientific Reports. It's titled Trial by Trial EEG-based BCI for Distress versus Non-Distress Classification in Individuals with ASD. So we're very excited about this topic because we have a lot of our DSI-24 and DSI-7 users are working with people who have on the autism spectrum, and we thought that this topic could be very interesting for you. So we invited him, and he was kind enough to agree to present some of his work with us. Dr. Akcakaya's work is focused on statistical signal processing and machine learning, specifically with applications to arrays of signal processing and non-invasive EEG, and as well as brain-computer interfaces. He also does some physiological signal analysis for health informatics. So his EEG work includes work on the detection, assessment, and rehabilitation of stroke-induced visual neglect using augmented reality in EEG, as well as EEG-guided electrical stimulation for haptic interactions in simulated environments. Today, he's going to be talking to us more about biologically informed interventions for emotionally dysregulated adolescents and adults with autism spectrum disorder. Dr. Akcakaya has over 100 peer-reviewed publications and is well regarded in this field, so we're very honored that he has agreed to do this webinar with us. With that, I hand it over to you, Murat. Thank you.
Thank you, Walid. Thanks for the invitation, and it's a pleasure to be here virtually. And hi, everybody. Thanks for joining us. And as Walid said, the title of my presentation is the Trial by Trial EEG-based BCI for Distress versus Non-Distress Classification in Individuals with ASD. So before I move on with my presentation, I would like to acknowledge my collaborators and the team members, and none of this could have been actually possible without their contributions. So Karla Mazevsky is our clinician and also the person actually who developed the autism rehabilitation intervention, and you will actually just hear about that soon. Karla is from Psychiatry Department of University of Pittsburgh. We also have Caitlin Connor from Psychiatry Department of University of Pittsburgh, Susan White from Psychology Department of University of Alabama, and we have our my students Safa, Bishra, and Nathan from Electrical and Computer Engineering Department of University of Pittsburgh, and myself. And we have more collaborators, but those collaborators are mainly for the future part of the work, and for this specific paper or for this specific presentation that's related to our paper, this is our main team members. So the lifetime societal cost of autism spectrum disorders have been estimated to be around 3.2 million per individual. And there's a steadily growing population of adolescents and adults with autism spectrum disorder. And especially the loss of productivity and adult care are the largest contributions to this cost. So many youth with autism spectrum disorder, ASD, decline in functioning during the adolescence. And this decline is at least partially related to impaired emotion regulation. So the emotion regulation, ER, is defined as one's ability to modify their arousal and emotional state to promote adaptive behavior. So impaired emotion regulation, often evident and early in ASD, it may get actually worse in adolescence and due to increasing social demands. So therefore, the ER impairment contributes to negative social outcomes, and they lead to failure to learn new social skills and a cycle of social avoidance, and eventually a poor quality of life and lower independence. So the figure here demonstrates the summary of the propensity for impaired emotional regulation in autism spectrum disorder and its common manifestations and the resultant poor outcomes. So recently, there has been an increasing emphasis on developing evidence-based approaches to improve ER, emotion regulation in autism spectrum disorder. And my collaborators, Carla and Susan, they are one of actually the pioneers about this. They developed this treatment methodology, therapeutic like clinical method. It's called Emotion Awareness and Skills Enhancement, EASE program. And it's actually, it's a 16-week program, and it's basically developed mainly for verbal adolescents and young adults with ASD. And it's mainly to improve the ER capacity. So, and their initial results, this is still like an ongoing clinical study. And this data that I'm going to present today, the EEG data, is also collected through that study, EASE study. And their preliminary results show that this EASE study that includes mindfulness exercise shows promising results for the emotion regulation, emotion regulation. So the behavioral intervention techniques such as EASE that's developed by my collaborators are the most effective type of therapy for autism spectrum disorder. But the rate of generalization of such techniques to real life beyond clinical gains is in average only around 48%. And moreover, they are time consuming and costly. So to overcome this, there's a growing interest in complementing such clinical approaches with various low cost and easy to access technology-based tools. And this is mainly to improve the therapy efficacy without additional clinician time. And such technology-based tools were also shown to help people learn, or the target population to learn skills of functional living and social communication and interaction. And they are mainly used as self-learning and self-training tools. So these type of actually tools, technology-based tools, can be categorized into three items here as we are presenting, summarizing under 3 items. One of them is the first one is the video modeling intervention tools. So these, as the name suggests, they use videos for the video modeling behavior. So these do not have interactions and there is no feedback. It's just watching videos and learning from the videos. Because of that, it was shown that these type of intervention tools, technology tools, have moderate effects on enhancing social and communication skills. One level up is the computer-based simulations and games. And such tools actually have certain interactions and certain feedback. But however, still it was shown that they lack the generalizations to generalization of these skills that are learned through these games or the simulations to the real life activities, because there is no immersive experience, or there is no ecological validity. So the last item that I'm going to actually mention as part of this technology-based tools is the virtual reality-based tools. And these tools, on top of the computer-based simulations, they provide ecological validity and provide immersive experience. However, these are still new, and there is currently no evidence for generalization of skills learned by the individuals with ASD to activities of daily living. So this is basically the introduction to the ASD plus the emotion regulation plus the technology-based tools. So what we are envisioning as a team is that we would like to improve ER for individuals with ASD. And we would like to develop a system that's portable, cost-effective, and it's going to actually have high temporal resolution. It means that actually we can intervene very quickly. And then it's going to have a measure of emotional regulation that can be generalized to activities of daily living. For this purpose, so we would like to develop an EEG-based brain-computer interface, and then tightly integrate this to the EASE clinical treatment protocol developed by my clinician colleagues. So the overall idea is that what we aim to achieve is the improved emotional regulation, and accordingly decreased psychiatric symptoms such as depression and anxiety, and decreased problem behaviors, including aggression, and decreased functional impairments across settings such as school, work, home, community. So it means that improved independence in life. So that is our vision, and to achieve this vision, as I said, we would like to actually develop an EEG-based BCI, and we would like to tightly integrate this with the clinical treatment strategy, the EASE. So our envisioned system here includes a virtual reality headset, an EEG headset. And then through the virtual reality, we would like to present certain, for example, social interaction scenarios that can actually have changing levels of difficulties. And then it's basically going to challenge the user, challenge the target population. And then during those interactions, social interactions on the presented through the virtual environment, the EEG is going to monitor the brain signals. And then what we would like to achieve is that we would like to see a change in the distress level through the EEG, and use this as a feedback, for a guidance, for a feedback that we will actually present to the user, the target population. And this feedback is going to actually include the treatment strategies that are presented as part of this EASE program within the clinics. So overall, tightly integrating this means that in this technology-based tool, we are also reminding the users what they learn in the clinical environment, and we would our aim is to help them to generalize these to activities of daily living, hopefully through these scenarios. So our overarching goal is actually to use the VR and social interaction scenarios, but in this paper I will actually start with a computer game, and that is basically a card game, and I will actually explain what that card game includes, how to actually control this, how we are aiming to increase the stress level, and etc. So yeah, our overall idea is to identify brain responses to distress, and it's going to be our foundational step towards this BCI, development of this BCI. So as I said, I will actually show our preliminary results about this to achieve our overarching goal. And these preliminary results include EEG collection from a card game that's based on Affective Posner task. And the EEG data is actually collected by the SI24 system. And the electrode channel locations are listed here, and in addition to the reference location. And we collected data from 21 participants, and each participant was actually young adults. So we basically had to get their parents' consent. And these participants were recruited as part of this EASE clinical treatment program. So there are some more details here. So in terms of the seating the participants and everything was, at this point, everything is on the computer screen. So this is one of our, in the picture, you can see one of our research assistants. And this is one of the pictures actually we use to, demonstrate the system to the potential participants. So they sit at a table across from the computer, and they play a card game. And this card game depends on the effective Posner task, and it includes deception component. So every participant actually receives $25 for the participation. But there is actually, since this is a card game and there's a deception component, there's an incentive for the individuals to actually continue with playing. And that incentive is additional $50 if they actually win the game. But we design actually the game such that even though it includes deception, at the end, everybody wins the game and everybody actually gets this money, but they do not know that when actually they start to play the game. So this game is based on this effective Posner task, as I mentioned, and the entire paradigm includes multiple games. There are 5 games. It's based on the same game, based on the same card game, but with actually different deception components. So this card game includes these components. Here, there are 2 white rectangles here, or squares, and there is a star under one of these squares. And the player is actually is trying to identify where that star is located, under which square. And there is a blue square that's actually placed on one of those white squares as a cue or as a clue for the participant. So 75% of the time that blue square is placed on the correct card. So, and then basically the player chooses the card, and then if they choose the correct one, the feedback, visual feedback is like correct, you are correct. If they are wrong, you are wrong. If they are slow, the feedback says you are too slow. But the game 1, the initial game, only includes the correct and the incorrect decision. It means that we don't actually give any feedback about too slow because we do not know their speed. We use game 1 to learn their speed, and then we design game 2 based on that, also to present actually results in terms of being too slow. So the game 1 and game 2, they do not include any deception. And we do not actually collect EEG during game 1 and game 2. It is mainly to introduce the participants to the games and train them basically how to play this game. So then game 3 is when actually we start to collect the EEG. And actually just before game 3, there's a rest state before game 3. And game 3 is repetition of game 2 that includes, correct decision feedback, incorrect decision feedback, and too slow decision feedback. But different than game 2, it includes also a deception component. So what the deception component includes is 60% of the time, independent than the participants' actual response, we say that they are too slow. So this is mainly, this deception component is designed to increase the distress level. So game 3 includes that deception. So before game 3, there's a rest state of EEG 2 minutes, and then there's game 3, and after the game 3, there's another rest state, and then that's followed by a short audio-based mindfulness exercise, and then there's another rest state after the mindfulness exercise, and then there's game 4. It's exactly the repetition of game 3, and then after game 4, there's another rest state, and then after that, there's game 5. Game 5 is actually the cooling down period. It's the repetition of game 2. And this is when we make sure that actually during game 5, we make sure that everybody wins. And then they just leave as a winner. So this entire experimental paradigm is designed to increase the distress level. And what we are going to be looking at today is the win and the lose conditions and how the EEG can separate those 2 conditions and how they actually compare to the rest state. By lose condition, we basically consider both the too slow and wrong responses. So for that purpose, we first, after we actually get the data, we just bandpass filter it, and the bandpass filter frequencies, cutoff frequencies are at 0.1 and 40 hertz. And then after filtering, for the specific, like EEG specific to the events, to identify those, we get the EEG, 3 seconds of EEG, time locked to the visual presentation of the feedback. The visual presentation of feedback is, it's basically this correspond to this feedback screen, which includes like the correct, incorrect, or the too slow responses. So basically, we are just we are extracting 3 seconds of EEG data, time locked to those visual feedback. And then based on this extracted EEG, we are gonna extract certain temporal and spectral features. And in this study, we are gonna focus on 3 channels. And but this is basically, these are our preliminary results, focusing on the 3 channels, the frontal channels. And these, the selection of these channels are inspired by from the literature. So in addition to this, we also extract the same features from the same channels for the baseline. And for the baseline, we consider the 2 minutes the rest EEG, 2 minutes rest EEG that was recorded just before game 3. And it was 2 minutes, so we basically extract the same length, 3 seconds of EEG data, and non-overlapping 3 seconds windows. So that corresponds to 40 trials is our baseline. And so this slide presents our spectral and the temporal features. So as I mentioned, we focus on 3 channels, the frontal channels, F3, FZ, and F4, and we calculate the total power in these channels. And then we look at the power difference or the frontal asymmetry to identify the frontal asymmetry. We look at the absolute power difference between F3 and F4 in the frequencies of 4 to 30 hertz. And then we also calculate the theta power, total theta power, total mu power, and then total beta power. These are in addition to the temporal features that we consider, the P300, P200, and FRN, feedback related negativity at around 200. It's what we listed here as and 200, and LPP, late positive potential. And as I said, we use these temporal and the spectral features. We extract these from 3 channels, and we apply machine learning to solve 3 classification problems. One of them is win versus rest. The other one is lose versus rest. And then the last one is win versus lose. And it's not just that basically we apply the machine learning, but we also apply channel and feature selection. And for that, we use weighted sequential forward algorithm. So what we are interested in is what is the most informative channel and what's the most informative feature for these classification problems. So this is the first results slide. So this slide actually shows the accuracy, sensitivity, and specificity and F-score for the classification, 3 different actual classification problems. Especially win versus rest and the lose versus rest, they have very high accuracy, specificity, and sensitivity, and very high F-score. So for the win versus lose, our accuracy and sensitivity, they are high, but we actually have some problems with the specificity, and there is some decrease in the F-score as well. So what we are showing here is that it's easier to separate the win from the rest and the lose from the rest. It becomes more difficult to identify the difference between the win versus lose looking at the EEG. So that requires actually, even though these are like promising results, that still requires some more work from our end. And that slide actually shows which features contribute. As I mentioned, we use the feature selection algorithm, and this slide shows which features contribute significantly to the classification problem. So for the win versus rest, we see that P300 feedback related negativity and the total power in the frontal channels, they contribute the most. For the lose versus rest, we see the late positive potential, P300 and FRN, they contribute the most. And for win versus lose, we see the power in the frontal channels, the total power in the frontal channels from 4 to 30 hertz, and P300 EEG features actually they contribute to most, and also FRN, LPP, and the power in alpha they contribute equally with some considerable contribution. So this slide basically summarizes which features are more informative about which classification problem. And then the last result slide that I'm going to actually present is to validate the significance of this feature selection. Because how do we know that actually we are not getting any random effect from the selection of the features? So in order to show that actually this feature selection doesn't contribute randomly to the classification performance, what we did was we applied a permutation task. So for each classification problem, for each individual, we just assigned, instead of through selection of the features, we just randomly assigned the features for each individual and performed the classification and extracted accuracy and F scores. And we applied this permutation multiple times, and we are actually showing the average or the mean value of the accuracy and the F score in addition to their standard deviations here. There's a significant decrease in the accuracy and F score for all the classification problems when we apply this permutation task. So this is kind of a validation to show that the feature selection is not actually random. There is information that we actually gain from that feature selection. So in conclusion, so we developed this system as a first step. This is our preliminary results, but it's a promising step towards identification of distress and non-distress conditions in individuals with ASD. So we showed that actually there are certain features that we can extract from the EEG that can contribute to trial by trial classification of actually different events, especially win versus lose, win versus rest, and lose versus rest. And different actually features contribute in different amounts for different actual classification problems. But overall, what we noticed from here is that P300, LPP, and then FRN, or no, not FRN, but the total power, total frontal power, they were the most common features that are selected across individuals, across all three problems, and they need further attention for the future processing. So our future work, as I said, this is our preliminary step, and we have, I described also the vision of what we want to achieve. As a future step towards achieving our goal, the first step is going to include the effect of the short mindfulness on different actual rest states. That's what we are working on right now. And then we are going to actually investigate game 3, game 4, and game 5. The results that I presented right now, they only rely on game 3, the first game with the deception. And we are going to actually look at game 3, game 4, game 5, and potentially the propagation of the features that are extracted that are most informative about distress, non-distress levels, and how they propagate during these games. That's what we are going to actually investigate. And the reason for that is actually because we want to build a real-time system that will monitor the changes in the distress level based on EEG. So that's how we are trying to achieve that. And we call this actually real-time monitoring as the phase 2. And the phase 2 is gonna basically use the same card game, but provide feedback and integrate the clinical components based on the monitored EEG. And that is the phase 2. And hopefully for the phase 3, we are aiming to integrate the virtual reality and include the card game plus further social interaction scenarios for the envisioned system. So this is the end of my presentation. Thank you for attending, and I will be happy to answer any questions.
Thank you very much, Murat. I'm sure you're hearing everybody's applauding in the background.
Thank you.
We received quite a lot of good questions, and I was sorting them through and organizing them while you were talking. So I will give them to you in an organized fashion. And for people in the audience, please feel free to keep asking more questions in the chat. I'll add them as we go along. The first set of questions that we have relate to the classification. You mentioned that you used the mu rhythm in your data set, but in your results we didn't see any features being selected that are mu rhythm features. Also, we were curious. Some people were asking about the SMR and whether you looked at SMR. Those two, I understand, are typically related. There are people that have shown some relationship to autism. So what did you find with the mu rhythm? And
so the mu rhythm we calculated as the feature, but it was not actually selected as part of the, with our limited number of participants, 21 participants, and with the the classification or the selection algorithm that we used, mu rhythm didn't show up in the top. But it doesn't mean that actually, because we are also in the process of, collecting more data and investigating different channels, especially just consider the fact that we focused on the frontal channels here, 3 channels. Maybe once we actually start to include more channels in the analysis, the mu rhythm is going to actually start to show up further, because you expect to see them actually a little bit more towards the, sensory-motor regions potentially. So we still actually do not ignore that could be the possibility. It may actually start to show up when we start to include more channels in our analysis.
Okay. Yeah, it's also possible that the SMR, and some people have shown that that's more related to interaction with people. So maybe when you go from the card game towards the interactions and more real life scenarios, there may be some interesting effects to be seen there.
Yeah.
That's there was a short question, why 3 seconds only? I presume they're asking about the duration of your epochs and classification. Is there a reason why you're using 3 seconds of data as opposed to longer segments?
So the 3 seconds of data is because of the paradigm. We just assumed, because we need to actually keep this in a certain amount of time. So the max or the minimum amount of time, that's basically starting from the visual feedback to the next trial, is 3 seconds. So based on our paradigm, that's the minimum amount of time. And then we actually add some random plus on top of it. So, and we are trying to actually keep it in a reasonable amount of time, because if we actually increase the length of the trials, each trial, it's going to end up it's going to actually increase the entire experimental session. And that is the reason. The paradigm is the reason why we are actually focusing on 3 seconds. And the reason why we are actually focusing on 3 seconds is, just we wanted to keep it short. And 3 seconds is actually a reasonable amount of time also to calculate the spectrum for the EEG.
Perfect. Thank you for that. Another question was with relation to correlating to the predictors. So I'm interpreting this question to mean that the question is, and please correct me in the chat if I'm misinterpreting your question. Did you classify the subjects based on their levels of the spectrum, where they are on the spectrum? That could be a way to look for correlations, I presume, or the correlations might be to look at the relationship between the response intensity and the EEG markers. So did you look at either to look at the level of intensity of their stress and look at correlations with EEG, or did you look at where they stand on the level of the spectrum?
So that's a very good question. Thank you. So the level of the spectrum, so all these participants actually, they qualified for this EASE study, as I was describing at the beginning, and the monitoring and the evaluation was done through this ADOS 2. And we didn't actually look at the level of the intensity of the autism or the severity of the autism and how they correlate with the performance of the EEG, or their like how we actually identify the distress from the EEG. We didn't look at it yet, but it's actually part of our future work for sure. In addition to actually, my collaborators, they are developing, they developed a new measure for the the intensity for the autism, and we are going to be actually investigating that as well. In terms of actually the classification, so the classification right now purely depends on win versus lose and how we interpret like lose and win. And we just said that like, okay, lose, let's say that it corresponds to distress, and win is the non-distress. So, and that's how we are actually looking at it. And after every game, we just basically ask participants to evaluate their, how positive they feel and how negative they feel. But we don't actually, unfortunately, we don't have this for every trial, because that is actually a long and lengthy process to get their evaluation for each trial. We cannot get a ground truth for each trial. Our ground truth right now assumes is based on the game outcome itself. So because
Sorry, that leads very nicely to another question, so I'll jump to that one, and that is, have you considered using additional input modalities to determine their stress levels, such as galvanic skin response, GSR, or heart rate variability, and perhaps those could be used as ground truth to help for your classification?
That's a very good point. We are not using them right now, but it's feasible to use them. And especially my colleagues, my collaborators, they have studies already in terms of electrodermal activity and the heart rate variability, and how they can actually use those modalities to predict certain unpredictable behavior from the individuals in autism spectrum. And it's potentially useful to use those, but we are not using them right now.
And so correlated to that, I'm going to bring another question that was asked, which was, are you using any behavioral assessment or eye tracking or video recording during those tasks, and have you analyzed that data and do you see anything in there?
So we are recording the sessions, and then, as I said, we also have just like the personal subjective responses of the individuals after each game. But the most important thing is that actually we have the videos. And as I said before, just like our aim is actually to real-time monitor the EEG and see if there's actually a specific feature or specific channel that actually shows some kind of like outlier behavior for each individual during this like the game. And can we actually correlate it? Maybe if we can correlate them with the videos, especially like, and if we see some kind of outburst, some kind of like different behavior from the participants, and we can actually correlate them to the EEG, that might be interesting. But it's still under investigation right now.
All right, thank you. Another question that came in, this is now in relation to sort of multimodal sensing, so I'm grouping that here, is have you considered using functional near-infrared spectroscopy, so FNIRS, as another signal source to detect blood flow through the prefrontal cortex to show and demonstrate focus or attention or other metrics?
So we didn't, my collaborators are doing that. But FNIRS is a good approach, but it's actually not as fast as EEG. So, and the blood flow, what I believe is, just correct me if I'm wrong, this depends on the similar bold response, and it's in the order of like around 10 seconds and so on. So you don't actually get response that quickly that you can see in the EEG. So that was our overall idea that can we actually see a quick change and quickly intervene. And the FNIRS might be useful, and I'm not against it. It's just like I didn't use it. I'm not very much familiar with that, but it's a potential way to look at it.
I'm going to help you share a little bit of information from what we do with FNIRS in relation to that question. So I think this person might have been asking about the use of FNIRS as a parallel measure to demonstrate focus and attention while you're doing the task. So we have recently developed an EEG plus FNIRS device, and we had a webinar about that. So if you're watching this on YouTube, you can click through the other webinars and see the FNIRS and EEG. So you can measure EEG and FNIRS at the same time so that you can apply some of the analysis that Murat is describing. And you can also do the FNIRS in parallel to look at mental workload and see, what they're thinking about. And so indeed, our EEG plus FNIRS is combined in one pod, so you can see the EEG and FNIRS at the same time. And you could certainly, we have gauges that interpret the FNIR data and the EEG data and provide you measures of mental workload and attention. That's certainly something you could do, and the two systems do not interfere with one another.
Perfect.
So next question, it relates to the EASE protocol, so a little set of questions here about that. So the first one is asking, what types of feedback are provided in case of stress by the EASE protocol? Are those positive or negative feedbacks? Well, what are the types of feedback that can be provided?
So that is basically the feedback that we envisioned is the strategy that they use here. So I guess it's very tough to see. It's the awareness, being aware of their emotions. It's the ABCD approach here that if you guys can see my screen. So I'm not the expert on that. My collaborators actually developed it, and I'm not a clinician, but from my understanding is that it's all about mindfulness, being mindful about everybody's emotions, and it's the first step, awareness. Breathe, change, and distract. So this is the main ABCD of their therapy, the EASE protocol, as I understand, or as I understood it. And this is what we were actually aiming to present them, or some variation of this, to remind these individuals how they were applying these strategies, ABCD of the EASE treatment.
All right, thank you for that. And the follow-on question on that is, how do you foresee implementing your closed-loop vision? Is this something that will be done in the clinic? Is this something that will be done at home? Will this be available to participants of different age, different ASD level? Will this work for participants who have severe ASD and are low functioning, so do not have speech? So I'm grouping a number of questions all together here. How do you envision being used?
So these are all good questions. At this point, I think the initial step, even though we envision this system to be going outside the lab environment, everything we need to actually first show that everything works in a lab environment with the target population. So, and the lab that you see is actually, this is where we do our stuff and where we get actually our participants. And so right now, the population that or the participants that we have, they are verbal participants in the autism spectrum. But the severity of the spectrum might or might not affect the usability of the system. We didn't investigate it yet, and that needs actual further investigation. Plus, this being used after it's developed, this being used or being widely available depends on, the outcomes of this project plus certain collaborations, because we do not develop the systems. We do not develop the hardware. We just basically take the hardware, we just integrate them or synchronize them to each other, and then we do this signal processing and we develop algorithms. So being widely available, as I said, it depends on also the hardware that that's going to be actually used, and that's going to be actually publicly available, and that needs collaboration, such as with like companies like Wearable Sensing, for example. So the future steps might include something like that. Yeah.
Thank you. And I would say at the moment, from my understanding of the EASE protocol, and we can provide more information for if people want to have some more questions about the EASE protocol. The EASE protocol is being provided by a psychiatrist or a social worker, right? It's not the automated EASE protocol.
No, it's presented by a psychologist. Okay. So yeah, it's basically applied by a psychologist. My colleagues, Carla Mazepsky and Susan White, they developed it, and they have a team of psychologists and the clinical psychologists. They delivered this therapy.
Okay. Thank you. So the next question is saying, how was your experience with the DSI-24 compared to using conventional EEG? So sort of on the utility side.
Yeah, this is actually a good question, because we need actually with this population, with the target population that we are collecting data from, we want to actually keep the experiments very quick. It's just like the setup of the EEG time is very important, and DSI-24 it's a plug and play kind of headset. You just it's very easy to set up and very high signal quality that we were actually able to get, and also the shape of it and like how it looks, the participants actually like it, and it's easy to set up, easy to take out, and then easy to get like high quality signal. And for this type of actual study with target population, if you are working with participants, and the target population from participants, not just like the healthy participants in the lab environment, but from the target population, the easy setup and high signal quality and being highly portable, they are very important points, and the DSI-24 checks all those points, in my opinion.
Thank you, Murat. And yes, I would add to that we have a number of users in the neurofeedback space that are using a DSI-24 or other devices for neurofeedback therapy on patients with autism, and they report the same thing, that the comfort and ease of use of the DSI-24 combined with the signal quality really helps make the process easier. And with regards to the previous question, Murat, we very much look forward to working with you and your team to get your technology out to some of those neurofeedback practitioners who are always looking for new tools to help their patients who could use that. I think there's two more questions here. One is with relation to virtual reality. It's asking if there's any interference between the EEG equipment and the VR, and I'll answer that.
Yes, you go ahead, and because you guys have a system for that, already,
So again, for those of you watching this on YouTube, we have another webinar that's specifically dedicated to the use of virtual reality integration with the EEG, and we have certainly tested. We do not have any interference between the EEG device and the virtual reality. And watch all of that in the YouTube, or contact Cameron or myself at sales@Wearable Sensing. We'll be happy to provide you more information about that. I think the last question came in towards the end here. I wasn't able to understand it, so I'm just going to read it straight to you. How to analyze and extract useful information between channels when the early stages of the research subject data is not enough. So I'm guessing that they're trying to say if you don't have enough data before a baseline, how can you classify the stress versus no stress? I'm interpreting what they mean by that here.
So if we do not have enough data from the rest state, and how we, is that the question?
Yeah, how to analyze or extract useful information between channels when the early stage of the research, the subject data is not enough. So I think, yes, they mean that if there's not enough baseline data.
Okay, so I'm just, this is again, it's, I interpret this about like the generalization or the generalizability of the, the methods. So of course, when you actually have a small amount of data, and we didn't actually publish it until we get some good amount of data. It's why we have like 21 participants right now, and in the currently we have more than 40 participants, and we have the data from them. But again, looking at small data, I do not know. It's basically, it depends on like how many trials you have, how many, if you are just looking at within each participant or across individuals. If you are trying to do across individuals and population behavior, I think small data is going to be a problem, and it is related to the, the statistics. I guess that's what they are, what people need to, just, or what they are intending to use. So this is as much as I understand about the question, but I think it's related to the power or the inference power.
Okay, thank you. And I said last question, but there is here one more. We'll take this one. If your objective is to elicit or induce an emotional response to measure mental and emotional feedback and behavior responses, have you considered biochemical groupings that correspond to each emotion being expressed? This would aid in managing patient or user and therapeutic modalities.
Biochemical?
I'm not sure what
we did.
Groupings.
Biochemical, I assume that actually it's just some additional, measurement to infer the biochemical reactivity, but we are not doing anything like that at this point.
Okay. All right. Well, Murat, this was it. A good battery of questions. Thank you all in the audience for those really good questions. Thank you, Murat, for a great webinar. We really appreciate your time.
Thank you.
Forward to more publications on all the topics you discussed as well. And for the audience, we look forward to seeing you at our next webinars. Thank you all very much.
Thank you. Have a good night.
Everybody. Bye-bye.